Hello students, how are you? I hope you all are fine and doing well. In last video, I have discussed about the structure and function of flagella and cilia. Now in this video, I am going to discuss some more structures present in the cytoplasm of cell, that is centrosome and centriole. And I will also discuss about microbodies. So let us start the class. So students, today I am going to discuss about some more structures present in the cytoplasm of cell. The first one is centrosome and centrioles. Now these centrioles are exactly small microtubular structures. As you remember, I have discussed about these microtubular structures or cytoskeletal structures in previous videos. So centrioles are exactly small microtubular structures, and these are present in almost all यूकैरियोटिक एनिमल सेल्स. सारे यूकैरियोटिक एनिमल सेल्स में ये प्रेजेंट होता है. दे आर यूजुअली एब्सेंट इन प्लांट्स, स्पेसिफिकली इन एंजियोस्पर्म्स एंड हायर जिम्नोस्पर्म, बट दे मे बी प्रेजेंट इन प्लांट्स, बट स्पेसिफिकली इन दोज प्लांट्स विच हैव फ्लैजेला. मीन्स ऐसे प्लांट सेल्स जिनमें फ्लैजेला प्रेजेंट हो, वहाँ सेंट्रियोल्स प्रेजेंट हो सकते हैं. Like in bryophytes, in pteridophytes, in cycads, etc. And you know, these centrioles used to duplicate or replicate like DNA during cell division. जब सेल डिवीज़न हो रहा होता है उस वक्त DNA जो है वो रेप्लिकेट होते हैं या डुप्लीकेट होते हैं. वैसे ही सेंट्रियोल्स जो हैं, सेंट्रियोल्स ऑल्सो गेट डुप्लीकेटेड और रेप्लिकेटेड ड्यूरिंग द सेल डिवीज़न, and they have ability to form basal bodies. Now what is centrosome? Exactly, centrosome is made up of two centrioles which are present at the right angle to each other. Means centrosome दो centrioles का बना होता है जिसमें वो दोनों centriole एक दूसरे से right angle पे present होते हैं. Now if we will talk about the structure, internal structure of centriole, then you can see in the diagram that the centriole consists of a whole of 9 peripheral fibrils similar to the flagella and cilia. जैसे कि flagella और cilia में 9 peripheral fibrils थे, वैसे ही centriole में भी 9 peripheral fibrils present होते हैं. But there is one difference. The difference is that in flagella and cilia there were 9 peripheral doublet fibrils. But you can see in the diagram of the structure of centriole that it contains 9 peripheral triplet fibrils. And from outside to inside, these 9 peripheral triplet subfibrils are named as C, B and A. Means जैसे आप डायग्राम देखेंगे you can see that बाहर से लेकर अंदर तक जब हम इसे स्टडी करें ये ट्रिपलेट फाइब्रिल्स को, then we are naming them C, B and A. Now one more difference is there in the structure of flagella and cilia and centriole. In flagella or cilia there were 2 central fibrils, but in centrioles no central fibrils are present. So the arrangement of fibrils in the centriole is 9 plus 0. Since there is no central fibrils, that's why the arrangement of fibrils in centriole is 9 plus 0. A question is asked that what is the difference between the arrangement in the structure or arrangement of the subfibrils or fibrils in flagella or cilia and in centriole. The difference is that there was the arrangement of 9 plus 2, and in centriole the arrangement of fibril is 9 plus 0. Now if there is no central fibrils in the center of centriole, then what is there in the center? In the central part of centriole there is a proteinaceous mass which is called as hub, and this hub is connected with the peripheral triplets. जो 9 peripheral triplets हैं उनसे ये hub जो है, जो कि center में present है, it is connected with that triplet fibrils. And how it is connected? This hub is connected with the 9 peripheral triplet fibrils by radial spokes. Okay. Now, one more thing is present in the structure of centriole, and that is pericentriolar satellites. Now, these 9 pericentriolar satellites which are present outside the centriole, it functions as nucleating center during spindle formation and during the duplication of centriole at the time of cell division. Or in the simple language, you can say that these pericentriolar satellites which is present outside the centriole helps centriole during the formation of spindle and during the duplication of centriole at the time of cell division. Now the function of centriole. The first function of centriole is that it helps in cell division by forming spindle pores or microtubule organizing center, or you can say MTOC. As you know that spindle formation is very important for cell division, and this spindle is formed from spindle pores or from MTOC. The second function of centriole is that it gives rise to basal bodies which further forms cilia and flagella. Another function of centriole is that if it is present in sperm, then one of the centriole present in the sperm used to give rise to the tail of sperm. Means जो sperm का tail होता है, it is formed from the centriole. So it was all about centrosome and centriole. Now next structure present in the cytoplasm of cell is microbodies. Now what is these microbodies? These microbodies are small organelles which take part in oxidation process. Now exactly these microbodies are of two types. First is peroxisomes and second is glyoxysomes. What are these peroxisomes? Peroxisomes are single membrane bound structures which is found in both plant and animal cells. Means ये peroxisomes जो हैं, they are found in both plant and animal cells in the close association with endoplasmic reticulum, mitochondria and chloroplast. Okay, now these peroxisomes contains many enzymes like amino acid oxidase, alpha hydroxy acid oxidase and beta hydroxy acid oxidase, which are involved in the oxidation processes. Now in animal cells, specifically in animal cells, these peroxisomes takes part in oxidation process of various biochemicals. For example, if a person is consuming alcohol, then this alcohol is detoxified by peroxisomes present in liver cells. Means अगर कोई person alcohol consume कर रहा है तो alcohol में जो भी toxins है उसका detoxification जो है, उसका neutralization जो है, वो liver cells में जो peroxisomes present होते हैं, जो peroxisomes liver cells में है, वो ही उसका detoxification करते हैं. Now in plant cells, these peroxisomes performs photorespiration process along with chloroplast and mitochondria. This photorespiration process is a very important process which occurs in plants. We will further study about this photorespiration process in plants physiology unit. So peroxisomes present in plants performs photorespiration along with chloroplast and mitochondria. Now next microbody is glyoxysome. These glyoxysomes are present only in plant cell, and its most important role is beta oxidation or breaking of fatty acids in plant cells. So students, with this the chapter cell, the unit of life, is completed. In next video I will start the new chapter, the cell cycle and cell division. Till then, stay home, stay safe, use mask, use sanitizer and maintain social distancing. Have a nice day.